name is JP Lawrence. I'm a postdoc at UC Irvine, but I'm going to be presenting some work that I did for my dissertation at the University of Mississippi on differential responses of avian and mammalian predators to phenotypic variation in Australian brood frogs. But before I get too far into that, I want to make sure everyone's on the same page with regard to aposematism. Aposematism is warning signaling. This is bright or conspicuous signals that's meant to warn would-be predators of consequences of predation, because these animals have secondary defenses, usually in the form of distasteful compounds, toxins, or morphological characterizations. Now, while I imagine everyone in this audience is not intimately familiar with all the research surrounding aposematic signaling, you probably intuitively understand the concept, because perhaps the best example of aposematic signaling out there is that of a stereotypical wasp. This yellow and black banding is something that we intuitively understand as means danger, don't mess with me, you're going to regret it. And that's what these animals are all trying to say to their respective predators. Now, there's a lot of research surrounding aposematic signaling that's really cool, but I don't have time to cover it all today. So I'm going to be focusing on stabilizing selection. So if you're a predator and you come across one of these red frogs and you taste it, tastes awful, you quickly learn that red frogs are bad, you want nothing to do with red frogs whatsoever. What happens when a novel form pops up, like a green frog? You have no prior experience to that. So you would, it's expected that these novel forms are going to experience disproportionately high predation pressure. And as a result, be weeded out of the population because they're initially rare. Thus, stabilizing selection is going to stabilize the overall phenotype of that aposematic species and limit diversification within this group. Despite that expectation, in a number of different groups, including the heliconius butterflies, the dendrobatid poison frogs, and the motylid wasps, we see considerable within and among population variation in signal. So this begs the question of why. Why do we see such variation when evolutionary theory clearly predicts that it shouldn't happen? So I'm going to attempt to examine this using the Australian brood frogs in the genus Pseudophrony. These are an Australian endemic genus, and they're called brood frogs because they have this really neat behavior where males will construct call chambers just under leaf litter, and they'll attract females to these chambers. The females will lay eggs, and then the males will guard those eggs until the chambers flood, and then they'll get out of there and the eggs will complete development. But what initially attracted me to these frogs is that they're super variable in color. Now, this variation is usually species specific, so you have some cryptic species, you have some conspicuous species, but it's a wide range of color variation. And while it is usually species specific, there are some neat species like Pseudophrony dendii, which shows considerable within and among population variation in color. Not only that, but some of these, all these species rather, have this really interesting character where their ventral surface is this black and white reticulated pattern. It's very, very conspicuous, and this is not as variable among species, although there are some, like Pseudophrony semimarmorata, which adds a little bit more flair to its signal. Now, these also produce alkaloids. They can either synthesize them de novo, or they can get them from their diet, like the dendrobatid poison frogs. So we have a system where there appear to be conspicuous signals, they have secondary defenses. This appears to be aposematic in origin, although no one's actually empirically tested this. So this begs the question of, are the dorsal and the ventral signals protective? Now at this point, I'm sure you're thinking that, well, yeah, maybe the dorsal signals are protective, they're very conspicuous, it's easy to recognize those. But when is a predator ever going to see the ventral signal? That's a really interesting point. These frogs have a really cool behavior in that when they're disturbed, they tend to freeze. And if you consider the sort of predators that they may be interacting with, something like a brush turkey might be scratching through leaf litter, might accidentally kick up one of these frogs. And when that happens, they sort of freeze. Not only that, but when they're really disturbed, when they're really poked and prodded, they tend to flip upside down and expose that ventral signal. So specifically, I'm interested in, are the signals protective against avian and mammalian predators? Now, to explain a bit about that particular question, we have to talk about the drivers of aposematic signaling. Aposematic signaling can be either with color, so if you consider the dendrobatid poison frogs with those really bright, conspicuous colors, or it can be with contrast. If you consider something like a skunk that has that black and white pattern, it's a very high contrast signal. But we need to understand a little bit about predator vision to understand why we need to consider these differences of color and contrast. So this is a very technical diagram of a retina. And our retinas are comprised of two different types of photoreceptor cells. We have our rod cells, which are light-sensing cells. They sense brightness. And then we have our cone cells, which are color-sensing cells. As humans, we have three different types of cone cells. We have a short wavelength cell that can sense in the blue spectrum, medium wavelength, which can sense into the green spectrum, and long wavelength, which senses into the red spectrum. Together, these allow us to see what we call the visible spectrum, that entire rainbow of colors. But if we consider something like a dog, which only has two different types of cone cells, it has very limited color vision. On the other hand, if we look at birds, they have four different types of cone cells, same as we do, plus an ultraviolet, so they can see into the ultraviolet. Together, we call these dichromats, trichromats, or tetrachromats, and while this is not the entire gamut of visual systems out there, this is a large portion of them. Notably, there are some exceptions and some extreme examples. Perhaps the most famous one is that of the Mantis Shrimp, which is a sexdecachromat. It has 16 different cone cells. Recent evidence suggests that they're actually pretty bad at discerning color, although some other people have some alternative theories, including being able to see a thermonuclear bomb of latent beauty. But since we are dealing with terrestrial animals, we probably don't have to worry about the aquatic ones right now. So if we consider animals that have these really bright colors and the visual systems that are out there, we can probably conclude that avian predators are likely important for aposematic signal diversification. And indeed, that's what a lot of the research has focused on, is specifically avian predators. I'm adding in mammalian predators for this particular study because of that ventral signal, that black and white high contrast signal that does not require good color vision in order to see and remember it. So pseudophony are found throughout Australia. They're largely restricted to coastal regions, and there are two main clades. There's the eastern clade and then the western clade. There is a weird individual species that's found right in the center of the continent I'm not going to talk about, but this work is focused on the eastern clade where the signal diversity is highest. So to conduct this study, we looked at how predators view novel signals, and we created clay models that were meant to mimic the different phenotypes found in pseudophony. So we have our brown model, we have one with flash marks, we have our yellow-headed model, our corroboree model, and our orange-headed model. And we also have our ventral models. Now to make these, what we ended up doing is getting a museum specimen, getting a 3D print of that, creating silicone molds, and then pouring melted clay, not chocolate, into these molds. Once the clay hardened, we pop them out, and then we paint them. And these were spectrally matched as best we could with commercial paints to what the actual frogs had. And this ended up resulting in creating a lot of models. These experiments tend to have very low predation rates, so we had to create hundreds of models for this. Now, at this point, I would like to thank my funding source for this, the NSF East Asia and Pacific Summer Institute's program that allowed me to go to Australia to do this research. So we created about 1,200 models of these 6 different phenotypes. We put them out on transects that range from 750 meters to 2.1 kilometers long, and we left them out for a week. This clay is nice in that it doesn't harden, so when it's attacked by a predator, they leave marks. So after this week, we would collect the models and examine each model to see if there are any teeth marks or beak marks left in the clay. We then examined or analyzed these results with a g-test of independence. So looking at our raw data, we saw this distribution of birds and this distribution for mammals. And while just visually this looks like there's some suggestive trends, I do want to emphasize that our attack rate was really low for these for this study. So we had too many treatments and not enough attacks in order to get enough statistical power to tell the difference between these. So to get around that, we grouped them into dorsal signals versus ventral signals and analyzed them in a few different ways. So first, we looked at all the local forms where we put these models and compared the dorsal signals to the ventral signals. And when we did that, we found that birds attacked the ventral signal significantly more than they attacked the dorsal signals. We didn't see any difference for mammals. When we look at all of the dorsal signals and compare it to the ventral, we see the same pattern. When we compare just the local conspicuous orange head form to the ventral signal, we see the same pattern. The same if we look at the novel forms that the predators have no experience with, we see the same pattern. So all this suggests that the dorsal signals may be deterrents to avian predators, and perhaps they're generalizing. I do want to add a massive grain of salt with that because we did have a fairly low attack rate. I can't conclusively say that these predators are generalizing, and that does warrant more research. It does appear, though, that the ventral signals do not deter avian predators from attacking these frogs. For mammals, however, we saw a little bit different results. The dorsal and ventral signals are equal. And by that, I mean they're either equally effective at deterring predation or they're equally ineffective. The design of this experiment wasn't able to discern which. But this represents the first assessment of the protective value of pseudophony coloration out there. So with that, I'd like to thank my collaborators, Mike Monty, Bryce Noonan, Kate Umbers, as well as my wonderful field assistant, Alira Jackman, as well as the Universities of Mississippi, Newcastle, and Western Sydney University. My funding sources, the Australian Academy of Science, the Australian Government Department of Education and Training, NSF EAPSI, and thank you guys for all attending. I'll take any questions. Yes?
Is there any evidence that all the color-colored polymorphisms are a form of on-signaling, so that sexual selection might be stronger than natural selection from this?
Yeah, so that's an excellent question. There's not yet. That's something that is on my radar that I would like to investigate. Color and toxicity in these frogs has largely been overlooked, but I think they're a really neat system that has a lot of research potential, and that's something that is definitely worth looking into. Yeah.
I think that mammals rely much more on smell than words. Do you think there might be an effect there?
Yes, so that is definitely a risk of these sort of clay model experiments, is that the mammals are primarily chemosensory based, and that could explain the results for that. We saw that there was no differences between the different forms. We didn't have a control set up for that, where we had like an amorphous blob of clay. That would be, if I went back and did further research on this, get a little bit more conclusive evidence, I'd include that so that we could see if a control was different at all from the actual models. Yeah.
Is there any evidence that attack rates vary across different microhabitats? Some places where some of these phenotypes might actually be a bit more
For this particular group, I'm not aware of that. For other systems, there is sort of local effects of predator communities. I don't know in particular about microhabitats. I'm not sure people have specifically looked into that. However, I definitely could see that being a potential interesting avenue of research. Yeah.
So I don't know much about the behavior of these guys, but I know in the bats they looked at like movement of models and gotten more stronger signals. Do you think that's something that could be worth looking at in the future?
Yeah, so the movement thing is a really neat aspect, and these frogs are very, very slow moving, so I don't know how much it would affect it, but it probably would affect it in some fashion. Great, thank you.